Okay, so we were talking about Mendelian patterns of inheritance. And remember that that is different from what the blending theory states. So I would put a little star in your notes about the blending theory versus the Mendelian theory, right? So the blending theory was what Mendel was working with, and he was trying to determine whether or not that was valid by using pea plants and looking at specific traits that the pea plants possessed and looking at how they were passed from parent to offspring. And he disproved the blending theory. So the blending theory is not upheld today because of the work that Mendel did and then subsequently all the work that all the other geneticists have done over the years to show how traits are passed from parent to offspring. Okay, so let's look at one example of here. So let's look at the seed color, okay? So notice that the seed color is either yellow or green. And remember that he picked these traits because they showed discrete variation, that there was no intermediate in the characteristic, okay? So if we look at this, and we did a cross of this, okay? I could give you the gene is gene Y. So this is my gene. And this Y gene exists in two different forms. So you could have the dominant form, which codes for yellow seeds. Let me change this so it's not hand. And only, okay. So you could have yellow seeds. And then remember the little y, which is the green seeds in this case. Notice I don't have a Y and a G. Notice that my dominant allele and my recessive allele are designated by the same letter. Because this is lowercase, sometimes you'll see them put a line over this. And so that means that it is underneath the middle line on a piece of paper. Okay, if you're thinking about the old-fashioned paper that you use when you're learning cursive. Okay, so these are alleles. And alleles are just alternative forms of a gene. Okay, so if we have a cross between a parent that is shown here, what is going to be the phenotype of the offspring produced? Are they going to have yellow seeds or are they going to have green seeds? Remember? Yellow, right? Because the uppercase Y is the dominant allele and the lowercase y is the recessive allele. So the offspring are going to be hybrids and they are going to have that combination of alleles, okay? So this would be yellow seeds. These are the hybrids. Okay. We don't need to do a Punnett square there because it's obvious that the offspring will get one dominant allele from one parent. So this could be like the ovules of one flower, and this could be the pollen taken from another flower, another plant. And the pollen was used to fertilize the flower of the plant that had this genotype. And then this was the offspring that was produced, okay? So then we sometimes have a cross where we cross two individuals that are heterozygous. So remember that heterozygous means that they have one copy of the dominant allele and one copy of the recessive allele. So these individuals are heterozygous versus being homozygous dominant or homozygous recessive, okay? So when we do this cross, this is sometimes referred to as a monohybrid cross. Why is it called mono? Does anybody know? Mono means one. Are we looking at multiple traits? Or are we looking at one trait? We're just looking at one trait, right? So we're just talking about a monohybrid cross would be a single trait, okay? So if we did this cross, then we might need to use a Punnett square, okay? So you want to remember how these genes are found on chromosomes. So this would be like, we now know, this would be the set of chromosomes that this individual has, right? And then this would be the set of chromosomes that this individual has, right? So these chromosomes are unreplicated, but prior to meiosis, which would be the production of the egg and the sperm in the pea plant, the chromosomes get replicated, right? So then you would get another arm put onto that chromosome. Those are actually called sister chromosomes. And then the chromosomes are going to separate, and they're going to line up, and they're going to separate so that you have the same probability of getting a dominant allele or a recessive allele appearing in the gametes, so appearing in the eggs or the pollen. So if we look at here, this is the male plant producing the pollen. We would have big Y and we would have little y. And that is actually 50-50. So it's kind of like a flip of the coin, right? And then the female would have the eggs, right? So that she would have the flowers and she's going to produce the fruit. And so there would be an equal probability of producing a Y, big Y, and a little y. Okay, does that make sense to everybody so far? Okay. So then I use a Punnett square. And the Punnett square is used to fertilize the egg and the sperm. So we just combine the combination of alleles. So big Y, big Y, big Y, little y, big Y, little y, and little y, little y. That's a Y, not a K, half of a K, okay? Okay, so what this gives us is the probability of offspring being one genotype or the other. So notice that 1 out of 4 is going to be homozygous dominant. So if I had 1,000 plants that were produced, I would think that about 250 of them are going to be homozygous dominant for that gene. Notice that about 1/4 are going to be homozygous recessive, so about 250 plants would be little y. And then about half the plants, on average, are going to be heterozygous, right? So this gives us the probability. So if we look at this, 1/4 is going to be homozygous dominant. 2 out of 4 is going to be heterozygous. And 1 out of 4 is going to be homozygous recessive, okay? So that gives us a ratio where we can actually get rid of that lower number, and it is a ratio called 1 to 2 to 1. And all that means is that 1 out of 4 is going to be homozygous dominant, 2 out of 4 is going to be heterozygous, 1 out of 4 is going to be homozygous recessive, okay? So this is what is referred to as the genotypic ratio. The genotype is the combination of alleles, right? So that's what the genes say. Just think, oh, that's got to be showing me the gene, right? So what is the phenotypic ratio? There's only 2 possible phenotypes, yellow and green. So there can't be 3 possible phenotypes. So what is the ratio? Does anybody, how many are going to be yellow? 3. And how many are going to be green? 1. So 3/4 or 1/4, right? That is the phenotypic ratio. Now, if I asked you how many are going to be green, you would say 1/4. You could say 25%. You could say that there's a probability of 0.25, right? So you could say it in multiple ways. You could say that there's a 25% chance of producing a green offspring or green seed offspring, right? So that would be a plant that produces green seeds. Oops, sorry. Or you could say that the probability, which is between 0 and 1, would be 0.25. So it'd be a 0.25 probability. Because probabilities, if it's 0, that's 0%. If it's 1, that is 100%. Okay. So I'll show you some multiple choice questions that could appear on like how I would ask it in a multiple choice question in a minute, a little bit. Okay. So has everybody followed so far what this kind of review? Okay, so now we have a problem here that if I'm looking at these individuals, I don't know which are hybrids and which are homozygous dominant. So I don't know which are carrying this recessive allele and which have 2 copies of the dominant allele. So I can do what is called a test cross. And so this is described in your book, okay? So this is my test cross. So a test cross is always an individual with the dominant phenotype. So actually, let me write it differently. So it would be this, okay? So individual with a dominant phenotype, we don't know if that's I put a question mark there. We don't know if it's a big Y or a little y. And we always cross it with an individual that is homozygous recessive, okay? So that's what a test cross is. So if this individual has another big Y, what are all the offspring going to be? Yellow, right? So you're like, oh, now I figured out that that individual must have been homozygous dominant because when I did the test cross, that gave me information, okay? If we look at the, what if it's a little y, then you'll notice that because it's a little y, then if we put it into the Punnett square, if this is a little y, right? Then there's a probability, a 50-50 chance that half of them will be yellow and half of them will be green, right? So we can use a test cross to determine what the phenotype of the unknown dominant phenotypic parent is. Obviously, we don't do this with humans, but we kind of do the same thing when we analyze a pedigree. So we'll get to that pedigree analysis in a minute. Okay. Oops. Okay, so let's talk about how these modes of inheritance as kind of discovered by Mendelian and developed more further by studying things like mice or studying things like fruit flies. So now we're kind of going beyond what Mendel discovered with his pea plants. Okay, so we can say that the inheritance is autosomal versus sex-linked. That's an L, L-I-N, linked, okay? So if it is autosomal, that means the gene is on chromosome 1, 2, 3, 4, or to the 22. So it could be on a chromosome, gene is on a non-sex chromosome. So it could be on the 17th chromosome or the 18th chromosome, but it's not on the X or Y, okay? So we say that that is an autosomal trait, okay? So like, we'll talk about cystic fibrosis. Cystic fibrosis is autosomal. It is on, I think, chromosome number 7, the defective gene, okay? Sex-linked is gene is on the X or Y. Okay. Now, which has more genetic material, the X chromosome or the Y chromosome? X. So it has like, I don't know, how many, 1,500, they found like maybe 1,500 genes on the X chromosome versus like 30 on the Y chromosome, okay? So what that means is most sex-linked diseases or traits are actually X-linked. So we could have X-linked, this is the majority, or we could have Y-linked. Now, sex determination is actually Y-linked because we know that there's the SRY gene that turns the reproductive organs into a testes, right? So sex itself is actually Y-linked. Right? If you have a Y chromosome and if all the genes are functioning, you will be a male by default, right? So you will always become, and actually not by default, but you'll be the male because it's present, okay? Okay, so that's one way we determine modes of inheritance. The other way is we could say that the trait is dominant or recessive. So dominant or recessive. Okay, so it's kind of interesting because most diseases are recessive. And the reason why they're recessive is that because they cause a big problem, that person will die and they will not be able to pass on their gene to the next generation. But if it's a recessive allele, then it can be masked. So it could be a person that is heterozygous and is a carrier for the defective allele passed on to the next generation. Okay. So the example that we talked about last week was what? Was a trait that was autosomal and it was also recessive. In our pedigree, what did we look at? Albinism, right? So albinism is autosomal recessive. Oops, recessive. Okay, so that means when I see recessive, when I see autosomal, that means I don't have to pay attention to whether individuals are male or female. Equal probability, it doesn't matter the sex. When I see that it's recessive, I know that big A is going to be normal in my little legend, and little a is going to have the trait, right? So this is going to have the lack of pigment, which would be albino. Okay. Another example, Huntington's disease. This is a neurological disease that has a late onset. So people don't know they have it, and then in their 40s or the 50s, they just start to lose their motor control. They end up in wheelchairs, and eventually they just even lose the ability to breathe, and their heart shuts down, all their organs shut down. So that is Huntington's disease, and it is actually autosomal dominant. And when I write it out with a big H, I could use big H. This is the disease, right? So now the big H is the disease because it's dominant, okay? So this is really good information to have when you're looking and you're trying to figure out a genetics problem. Is it autosomal? Is it recessive or dominant? And then the little h would be normal. So let's look at an example of this. So if I have individuals here, and let's say this person, and when I put a line through it, that means that they died of the disease or of the trait. So Huntington's disease caused that person to die. Now, is that person male or female? Female, okay? So these individuals are like wanting to know what their chances are of getting Huntington's disease. Actually, let me put some more information here. Okay, so let me erase that question mark. Let's see. Okay, there we go. Okay. So all these two individuals have it, but they haven't died of it, right? How could this person, if they have this awful disease, how could they have children? How could this person have children? What was the key about this disease? It doesn't happen until you're older, right? So you've already passed the gene on. So you've already reproduced. So you didn't know you were going to get this disease. until it was too late, until you decided to have children, right? So now there is the ability to determine, using genetic technologies, whether or not you have the disease before you actually get it. So if you have Huntington's disease, you can find out if you are going to get it. And it is your choice. So, you know, currently we don't demand that somebody know or be, this would be eugenics, but if you demanded that somebody find out before they were able to reproduce, or prevented them from reproducing because they had this defective gene, then that would be, you know, kind of against ethical considerations at the moment, right? So, but they have the option, right? So let's go through and figure out what these individuals must have had, okay? So what about this individual right here, which is individual 1, 1. So that's generation 1, individual 1. What must he have? Little h, and you're like, I don't get that because I don't understand, but you have to go back to this, right? Okay. So they're actually the homozygous recessive, okay? Now this individual could be big H, or big H, big H. But this is very rare, right? That is very rare. Because it'd be very rare, you would have to have 2 families that have this genetic disease getting together and producing, and so that is probably more likely that they're this. Okay, but it doesn't matter for our question. Okay. Okay, so this individual must have inherited 1 little h from his father, right? And 1 big H from his mother. So he got 1 little h from his father and 1 big H from his mother. So he has it. This individual doesn't have any history of it in his family. So you could safely put little h, little h. There's no family history. You could put no family history here if you wanted to. So then what's the probability that that person is going to have Huntington's disease and they're going to die early on of kind of a horrific disease? So we have to do the Punnett Square, okay? So, because there was 2 different answers there. So it would be big H, little h, right? That's 1 parent. Little h, I did the male on the side and the female above, it doesn't matter. Remember, I always put the big H's first, just for ease of reading, okay? So the answer is 50%. So it's like flip of a coin, right? So the question is, would you want to know if you were thinking about having a family, particularly, because you could pass that defective gene on. However, now, if this person found out they had it, there is actually the ability to screen eggs, or sperm for that matter, for the defective gene. So if this person was big H, little h, she could go in and have her eggs taken out and then screened to determine if the egg had the defective gene, and only use eggs that didn't have the defective gene. So she could theoretically have children that would not have the disease. That would be like 100% not have the disease through genetic screening. Are there any questions about that idea?
So, like, are you saying if they didn't inherit it, like if they got put on storage, they could have another child that has it?
No, I'm saying if they were, say they did the test and they're like, oh, I have it, I'm going to die early, but I still want to have children, okay? So they could actually have their eggs removed, and they could screen the eggs, and they could only use the eggs with the little h, and they would use sperm from the male that had the little h, and so you could ensure that it wasn't a 50-50, it wasn't a coin toss, that you could ensure that that person would have normal children. So you can genetically screen for diseases like this. Yeah. So that might be a reason to be genetically tested, but then you're like, well, is that eugenics? You know, good genes, that idea of eugenics, right? So I'll write that down, that word down. Right? This word became very popular during Nazi because they thought there was a superior race, right? So certain people with certain genes were superior to others. So that's a word, that's a group we've actually seen before in eukaryotic. So eugenics means good genes. And since, Hitler, it's been really viewed as negative, right? We should not prevent people from reproducing because of their genetics. Also, not too long ago, people were forced sterilizing people that had mental deficiencies. So if you were a person in a state hospital, you might, without even your consent, undergo sterilization so that you couldn't have children, so you couldn't pass your genes on to the next generation. And even people that were not really mentally deficit, but just people that they didn't think were, smart enough or good enough or of the right color, were also sometimes forced sterilized. So eugenics has a very bad history. And so screening has kind of this, everybody's like, oh, screening and producing the best, the perfect baby. This is just ensuring that a bad gene, a bad allele is not passed on. They want to have a healthy baby. Okay. Okay, so this is how it would appear. You don't need to write these down, but I just want to show you how, if I gave this on the midterm, how the questions would appear, okay? So notice that I give you the mode of inheritance. So this is cystic fibrosis. The disease has an autosomal recessive pattern of inheritance, okay? And then I say that it is in generation 3, individual 1, right? So you would know that I'm talking about, if this is generation 1, 2, 3, this is individual 1, okay? So then when I am doing these problems, I always give myself a legend, and you're gonna get more practice in lab today. So I say big C is normal. Little c is the disease. So cystic fibrosis in particular affects the respiratory system. So people get a buildup of mucus in their lungs, which lead to infection. And generally, up until most recently, people would die before they got into their 20s. They tended to die when they were like late teens, early 20s, because their lungs would just get so destroyed by infection. Okay. So then I would say, the genotype. Well, the genotype is the combination of alleles. So I have several genotypes here. Is B and E, are those genotypes? No, right? Because a genotype, we are diploid, right? We have 2 copies of every gene. We are not haploid. So those would be like gametes. So I can automatically say that's not them, okay? If I said phenotype, then it would be, are they normal or do they have the disease? But I'm talking about genotype. So what do you think the correct answer here is? D, okay? And I know that because if I look at my thing, it's got to be little c, little c. What about the parents of that person? What must they be? Big C, little c, right? Because they don't have the disease. Big C, little c, okay? What is that called, other than being heterozygous? What is that called? Being a blank for the disease. Carrier, okay? So they don't have any symptoms. They don't even know this disease even is in their family, right? But if 2 carriers get together, they can have a child with cystic fibrosis. So it comes out, right? So it just appears seemingly out of nowhere because they carry the recessive allele, right? Okay, so then I say, from the pedigree above, if individuals 2-1, this person and this person, have a second child, what will be the probability that they will have cystic fibrosis? 25%. You do your Punnett Square, right? So in this place, I would draw my Punnett Square. right? Big C, big C, big C, little c, big C, little c, little c, little c, right? Now I have to, before I do my Punnett Square, I would have to go back and I'd have to figure out what the genotypes of the parents are, right? And it's not, they're not always heterozygous, right? So you have to know the genotypes of the parents before you figure out the probability of the offspring. Okay, so that is how that is presented in a multiple choice question format. Oops. Okay. So here's another disease. So this one says it's autosomal dominance. Okay. Does it skip generations? Does it just like appear out of nowhere where the parents don't have it? No. So in autosomal, in dominantly inherited traits, there can be no carrier. So in order for the parent to pass it down, they actually have to have it. So there's no carriers, so it does not skip generations. So you'll always see that it has to be in one parent in order for it to be passed down, okay? So then I would give it a letter, so I could use A, right? This is the disease, yep. And this is the normal. Now I have to point out that Lou Gehrig's disease is only about 10% of it is inherited. So if you know somebody with ALS or Lou Gehrig's disease, it could be that it was an environmentally induced mutation. Okay, so not very many of them actually show a pattern of inheritance like this. Okay, so what does this person have to be? Actually, let's look at the people that don't have it. Okay, those people don't have it. These people probably don't have it, right? Because it hasn't shown up yet. Okay. So what must be the genotypes of those three that have it? Big A, little a. So they're not carriers, however. Why are they not carriers? No carrier is because they have the disease, right? They're not carrying the disease, they actually have it. So they have the disease and they pass it on to their offspring. And this is another one, just when you see these dominant lethal diseases, they are generally late onset, right? So that's why it's much more common to have recessive diseases than dominant diseases. Okay. Okay, so let's look at some Mendelian traits in humans. So we'll put in humans. And there's actually an online website, if you're interested, that is called OMIM, which is called, that stands for Online Mendelian Inheritance of Man. And we're going to look at some of these examples in lab today. Okay. So there are diseases that are caused by a single trait. So the characteristic, these are all caused, excuse me, by a single gene. So they could be traits or they could be diseases. Traits would be like having a cleft chin, right? So having a big cleft in the middle of your chin, that is a Mendelian trait, right? And also the ability to roll your tongue. So that's some phenotypes that are easy to look at, okay? Okay, so these are two different traits, and they are dominant. So if you have a cleft chin, you have the dominant version of the gene, and if you can roll your tongue, you have the dominant version of the ability to roll. But these are determined by two separate genes, right? So we have two separate genes, okay? So, for example, if we used C, C is where you have the, oh, excuse me. Yeah, C is where you have the cleft. Little c is no cleft. And notice how I put a little line over that? That makes it much easier for me to keep track. So I know that's a lowercase. Okay. And then let's put T is ability to roll your tongue. And little t is no ability. So some people simply cannot roll their tongue into a little tube. Okay. So I can look at two traits inherited together, right? So passed together. So these are actually, we kind of assume when Mendel was doing his work, he didn't know anything about chromosomes. Okay. So we're going to assume genes separate independently from one another. And this means that they are not on the same chromosome. If genes are together on the same chromosome, and if they're really close together on the same chromosome, then they don't separate independently. They're more likely to appear together, right? But we're going to assume that the genes are on separate chromosomes. So like cleft chin could be on, I don't know which chromosome is on. It could be like 2, ability to roll your tongue could be on 10, chromosome number 10, separate chromosomes, okay? So I can put this together, okay, these two genes together, and watch how I do it. So for example, I could have a person with this genotype. Okay, this is my genotype. What do they look like? They have a cleft chin and they can roll their tongue, right? Okay. But, oops, I forgot to put the little lines over them. No cleft chin, no ability to roll your tongue. Okay. So, in order to do a Punnett square, I have to figure out all the possible combinations that these two genes could come together in a gamete. And I purposefully did ones that are not too complicated. Okay. So I just want to show you that big C and big T could come together into a gamete. Okay. That is a gamete. So let's say that's the sperm. Okay. Notice that it is haploid because it has only one copy of each gene. Oops, sorry. Go back. Okay. What's the other one? Big C, little t. There's only 2 possible sperm. Okay. If that's the male. Okay. How about the female? What is the only possible combination? Little c, little t. Okay. So I can put these together, and when I do this, my Punnett square is pretty simple. It's just a 1x2 Punnett square, right? So I keep the C's together and I keep the T's together just for ease of reading. I always put the dominant allele first, okay? So I would put big C, little c, big T, little t. Okay, so see, I just combined this gamete with that gamete, okay? And then this one is big C, little t, little t. Okay. So this one is like one parent in phenotype, right? Because they have a cleft chin and they can roll their tongue, right? This one has a cleft chin and they can't roll their tongue. So this is an offspring that is not like either one of the parents. So that is how you do when you have the inheritance of more than one trait. Okay, so that's how you have two traits, two different letters, figure out possible combination of gametes. Okay. Now, in the most complicated form, you would have something that looks like this. Okay. So this is the most complicated Punnett square that we would do in here. And so this would be like this. And this is you going back to P plants again. Oops, big G, big G, little g. Okay. So if that's not a if the other one was a monohybrid, what is this one called? That's a big G. Monohybrid versus, what's this? What's two? Dihybrid. Okay, so this would be a dihybrid cross, right? And there's actually 4 possible combinations for each of them. So that gives you a 4x4 Punnett square with 16 possibilities. Okay, so that is a dihybrid cross. Okay. So I generally don't make you do those. This complicated of a one, but I think maybe you do that in lab today. I can't remember actually if I put one of those on there or not. Okay, I'm gonna skip that. Okay, so now we're gonna go to exceptions to Mendel. We could write exceptions to Mendel up here. So Mendel had a very basic system figured out, but we found that there was always like exceptions to the rule. Like he picked out such a simple system that we know that it's a little bit more complicated than that. And so over the years, we've come up with how there are exceptions to what Mendel discovered, okay? The first one is that sometimes we see what is called incomplete dominance. So if 2 alleles are on a scale here, right? And we have, let's see, complete dominance on the scale, complete dominance. So an example of that would be what we just looked at. So like Huntington's disease, completely dominant, right? If you have the bad allele, you will get this disease. It's completely dominant over the system. So that's Mendel's, all his traits showed complete dominance, right? At the other end of the extreme is what is called co-dominance. And this is where 2 alleles are equally expressed. And I'm going to foreshadow here. This would be like the AB blood type. We're going to do blood typing in lab today. And so the AB blood type means that you have the A and the B gene present on the red blood cells. Okay, so that would be codominance. So then we have this gray area here, which is called incomplete dominance, right? So that's the gray area. So some alleles are like a little bit less dominant, you know, so sometimes you get the expression and you can see the phenotypic change in an individual that is a heterozygous. So the result is there's a difference between the phenotype of a heterozygous individual versus a homozygous dominant individual. Okay. So now we can tell if somebody is a carrier. because their phenotype demonstrates it, right? Because we have incomplete dominance. The recessive allele is not completely masked, okay? So in this case, sometimes we use, instead of big R and little r, we use a 1 and a 2, okay? So let's look at flower color in 4 o'clocks. Okay. So in this case, we have big R, 1 is red. Big R, 2 is white. So 1 is not completely dominant over the other. Okay. So you could have this combination where you would have R1, R1, that is red. You could have R2, R2, that is white. But if you have R1 and R2, then you are pink. So I can tell the difference. I can say, oh, well, the heterozygous individual is a different phenotype, okay? And we're going to look at this specifically in a disease called sickle cell anemia, where you have a gene that codes for hemoglobin, and you have a mutation that has that is incompletely dominant, and that the individuals that are heterozygous for it actually have an advantage under certain circumstances, and it's selected for. So that is the example of incomplete dominance. Okay, so we also need for today's lab to talk about the idea of multiple alleles. So multiple alleles means that there are more than 2. Right? So you have not just a dominant and a recessive, but you can have more than you could have, like you could have up to 7 or 8, or I don't even know how many that they found in different genes. So you can have more variation in the genes than just 2. Okay. So a good example of this is our blood type. So we call this the ABO blood type. And weirdly, this is actually a gene that they have designated as the I gene. I'm not sure why it's called the I gene, but that's the letter that they gave it. And then the alleles are I with a big A, I with a big B, and then a recessive allele, which is a little I. So instead of having 2 alleles, we have 3 alleles. Okay. We still have only 2 copies, but we have 3 alleles in a population, right? But you only still get 2 copies. So if we look at the genotypes, instead of 3 different genotypes, we now have 6. So if we look at our genotypes, we have to figure out what are all the different combinations. So we can have this combination, right? So we could have 2 copies of that allele. We could have 2 copies of this allele. Or not 2 copies of this allele. We could have a big A and a little I, excuse me. And then you could have this one. You could have big B. I'm putting these in an order and a reason. Okay. Oops, sensitive, okay. Oops, sorry, okay. I hit there and not there, okay. So what's another combination? And then one more. Okay, so those are the genotypes. The phenotype is blood type. And it has to do with proteins that are found on the surface of red blood cells. So remember how we talked about some proteins embedded in the plasma membrane serve as cell-cell recognition. So it allows your cells to tell your cells from foreign cells. And so the reason why blood type is important is that if I'm going to get a transfusion, I need to know that I'm getting the right type that will match, right? If I get a mismatch, my immune system will attack the foreign blood and it will cause it to clump and cause me to die, possibly, okay? So this would be, these two give you type A. Now, if you're wondering about positive and negative, we're not getting there yet, okay? That's a different gene, okay? So this is type A. This would be type B. What do you think this one with the little a or little I is? O. And then this one is type AB. Okay. So this means that if I'm type A, I have A proteins on the surface of my red blood cell. If I'm type B, I have B proteins. If I'm type O, I have neither. The O means I have neither A or B. And then type AB means I have both. Okay. So which one of these, maybe that you know already, is what is referred to as the universal donor? O, right? Because it has neither A or B, that means it can contribute to all of those and it will not be detected as foreign. So this is the universal donor. So if you have type O blood, you might be called upon to donate your blood because it's useful, right? If you're type AB, this is what is referred to as what? Universal what? Recipient. So it's actually better for you to be, from your own not altruistic, but from your own selfish perspective, it's better for you to be type AB because you can receive anybody's type of blood, right? But if you're altruistic and want to be giving, right, you want to be type O. Type O can only receive type O because if it received any of the other ones, it would be detected as foreign. And the way that it detects it as foreign is antibodies would bind and cause the red blood cells to clump or to agglutine. Okay. Okay, so even though we have 3 different alleles, we still use the Punnett square exactly the same way, okay? So I go to, so you need to find a topic. Okay, so let's look at a person who is this. What are going to be the phenotypes of the offspring? I can use a Punnett square if I need to. So this, it doesn't matter, this is not sex linked, so it doesn't matter if it's on a Y or an X. This would be my Punnett square. This individual can only produce I. This one's going to be produced 50-50. Actually, that's just one line there. Okay. I drew my lopsided. There's just a 1 by 2. So it's going to be IA, little I, IB, little I. So interestingly, their offspring cannot be either like the mother or the father. Right? So their offspring are going to be either type A or type B. The parents were A, B, and O. Okay? Now we're going to talk about gene interaction in a little bit, so keep in mind that there are other genes that can influence blood type. So we'll talk about that, not today, but on Wednesday. Okay, so there is also, along with blood type, a positive and a negative. Okay, so I could be like O negative, or I could be O positive, or A negative, right? And so this is actually a separate gene. And does anybody know the letters that is designated for this gene? It's actually 2 letters. It's RH. And this is the gene. And it's after because it was discovered first in the rhesus monkey. So it was first discovered in the rhesus monkey, and that's why it's called RH. So if you are RH positive, right, you are going to produce, or you're going to have RH proteins on your red blood cell. Okay. If you are RH negative, you will have no RH proteins. Actually, maybe I should not use positive or negative here. So just no RH proteins on red blood cells. Okay. Okay. So this is determined by a gene, and sometimes it is used, they just use RH, but sometimes they use the gene D. Okay, so D is RH positive, and little d is RH negative. So positive is dominant, and having no RH protein is recessive. So what would be this genotype of this person? What would be their blood type? O positive. O because of the eyes, and that they have, this is the dominant allele, and it masks the recessive allele. Now, sometimes the reason why we actually pay attention to positive and negative could be due to transfusion, but it also has to do with a problem with pregnancy that can occur with pregnancy. And so in pregnancy, if a female is RH negative, so if pregnant female, this is humans, okay, and she has a RH positive fetus, okay, she may produce RH antibodies that cross the placenta and bind to fetal red blood cells. Okay. This can cause the red blood cells to be destroyed. And so it can cause damage in the fetus. So it can actually lead to a miscarriage. So if you are, and you know you are RH negative and you're female, then this is going to be, and if you ever get pregnant, this is going to be of concern to you. So if you're negative, and you're female particularly, and you have a baby, they will want to know if the baby, what the baby's blood type is. Because if the father has RH positive, then there's a good chance that the baby and the fetus will also be RH positive, okay? So then what they do is they give the female a shot, and they give the female a shot of a chemical called RhoGAM. Okay. And Rho obviously is RH, stands for RH. And GAM is I don't know what that means, but what this is a chemical that will bind. Okay. So this is a chemical that prevents maternal antibodies from forming. So it actually binds onto any fetal cells that cross the placenta and get into the maternal circulation. And so it prevents the mother from developing antibodies that then will attack the fetus. And so you have to have a special Rogan shot for that. And it's actually more dangerous in the second or after the first baby, because if you are Rh negative and you have an Rh positive baby, you might not be exposed to that baby's blood until the baby is born. But then the second baby, if the second baby is Rh positive, then you could really have a chance of causing a miscarriage due to the production of these maternal antibodies. So that's just an example of why Rh negative and Rh positive are in particularly concerning type of blood type. Okay, let's see what we have next. We might quit soon. Okay, we'll talk about one more thing. Okay, so epistasis is a term that is used to describe when two genes interact to determine the phenotype. So this is called gene interaction. So two or more genes interact to determine phenotype. Okay, and this is actually related to blood type. An example of this is related to blood type as well. So we have the H gene, and this is a codes for an enzyme that attaches proteins to red blood cell. Enzyme that attaches proteins to red blood cells. Okay. So H is the normal enzyme. Allele that is common in some populations is the abnormal enzyme. So it codes for the defective enzyme. Okay. So what this means is a person could have this combination. So they could have this combination of genes. Okay. So now we're going back to what we learned about the A. So if I was just looking at this, what type of blood type would that person be? AB, right? What about this? Oh, that is normal. I'm sorry, this is normal. Okay. So if I look back at this, this would be normal. Okay. So this would be blood type AB. Okay. So this is, sorry, AB. Okay. I meant to write this. That would be type O. Okay. So what I'm saying here is that the H gene interacts with the I gene. So if we have a defective enzyme, it doesn't matter what we have for A and B, no proteins will be attached to the outside of the red blood cell. And so a person would be type O. Okay. And so we do not use blood typing to determine paternity. So it used to be in the past, like if a male was like, I don't think that's my kid, they would look at blood types, right? And they'd say, oh, yeah, that can't be your kid, right? But for this reason, we can't use blood typing because look at this. If we have this combination, and I'm going to cross it with this person. Okay. Those two parents are AB. They should not be able to have a type O child, right? But if I look at the H, what is the probability that they'll actually have a little h, right? Ignore the I. Do I Punnett Square? 25%, right? So they can actually have an offspring. The parents are AB. They have an offspring that is type O, right? That type O is called the Bombay, I think that's a cool name. Phenotype. I think maybe, I don't know if it was discovered in Bombay. No such things Bombay anymore. They renamed it. But that's the person that is type O because of H, not because of I, right? And you look at that and they say, oh, that guy cannot be the father, right? No way that there is. Right? But if you take it into consideration H, right, then there is a possibility. So you could be type O, and you could actually have different I. And so your children could be completely different than what you would have expected in that particular instance. So they use DNA fingerprinting now, DNA genetic tests to test for paternity, which is much more precise than looking at phenotypes. Okay, I will stop there for today, and I will see you in lab this afternoon. So anybody came in late and needs a copy of the review sheet for the midterm?